Yeah, you have to go louder. Rebecca, that's quiet, so I hope so.
All right. Now we're starting. Okay. So chapter 17 is about transcription and translation. So a couple of things you need to remember from biology 1 is the flow of genetic information in the cell. So the nucleus in eukaryotes houses our DNA, and it's just hanging out in the nuclear region of the prokaryote. But the DNA is only copied before a cell divides. What happens the rest of the time during like G1 or G2? The cell's actually living and needs to use this DNA to make proteins and RNA when necessary. So it's not just hanging out there not doing anything. It's actually used a whole lot. And those processes are called transcription and translation. So proteins are the link between what we call genotype, which is your actual DNA, like nucleotide sequence, and phenotype, which are the characteristics that we have or display. Gene expression is the process through which the DNA is transcribed and then translated into polypeptide sequences. Just vocabulary you need to know. So the beginning of this chapter, and this might have been in one of those sidebars about experiments, was this information about how they actually knew that the DNA codes for polypeptides and all this fun stuff. For a long time, physicians, like doctors, were the ones that kind of had these first ideas, and because they had patients that were showing symptoms of metabolic disease. By looking at metabolic diseases, physicians were able to sort of say, all right, well, they have a problem with a specific, like they're not making a specific protein. And it's because they have a specific mutation. And it took a lot of time and research. But for example, there's a couple of things. Alkaptonuria, people, if you can imagine if you were going to like pee and then your pee is black, you would totally freak out, right? Okay. It's because they have this alkapton is a byproduct being produced. I have it on the slide. Anyway, CKU, phenylketonuria, is another type where these individuals are missing a particular enzyme and they can't break down phenylalanine. Every specific metabolic disease is caused by a specific non-functioning enzyme. And you can see down here, if your enzyme that goes from A to B is now functioning, you won't produce anything along down the line. But you can have a different one mutated. Anyways, here's a different metabolic pathway that you have phenylalanine, and eventually it's converted into carbon dioxide in water. So this would be normal. All right, but if you are blocking, or if you already have an enzyme that is defective, if you can't convert phenylalanine to tyrosine, then you're going to produce PKU, or phenylpyruvic acid, and you'll have PKU. And it's because you're missing that very first enzyme, phenylalanine hydroxylase. The genes are mutated, you don't make it, it doesn't work. People that have PKU just need to make sure they avoid having phenylalanine in their diet, because if they have it a lot in their diet, especially when they're young, it can cause like mental disability to develop. It's not so bad when you're older, but you just avoid it. Anyway, so if you have an enzyme, so the hydroxyphenylpyruvic acid cannot be broken down into homogistic acid, you'll have tyrosinosis, there's albinism and cretinism, different things. So tyrosine can be converted to melanin. If that enzyme is defective, then you'll have albinism, where you don't produce melanin, right? And you have no pigment in your skin or your eye.
That's vitiligo.
That's different. Yeah. And that vitiligo is it can function in some places and not others. You know, they have spots and things like that.
Like I did on.
Albinism is like in no DNA in any cell in your entire body is it functioning. Does that make sense? Anyway, so that's that. All right, so moving on. So for a long time, scientists just thought each gene coded for one enzyme. All right, so there's an experiment done by this guy named George Beadle and Edward Tatum in the 40s/50s, and they had this experiment where I'm just going to talk about it over here. They were interested in a fungus called Neurospora, and what they did is they created mutations. They understood at this time that if you expose something to X-rays, you're going to produce mutations, and they'll grow differently. They understood that. So what they did is they said, all right, and they understood enough about a particular metabolic pathway, the one that produces arginine. What is arginine? Arginine is an amino acid that's necessary for production of a lot of proteins, okay? They knew enough about this particular pathway to know that you need there are different enzymes in the production of arginine. And they also produce in the pathways this thing called ornithine and citrulline. These are sort of the precursors. Okay, they knew enough about all this information. So all right, let's grow this nurse spore, and we're just gonna X-ray the doo-doo out of it and see what happens. And so they had 4 different types of medium or agar they could grow their fungi on. The first type of agar over here on the left-hand side is called minimal medium, meaning in that media, in that agar, all that was there is something called this precursor. And if the fungus was not mutated or if it was normal, it had all the enzymes necessary to convert that precursor eventually into arginine, and the fungus could live because it had those enzymes that were functional and it could use arginine to make its protein. Okay? They also knew that sometimes, for whatever reason, if there was something going on with the fungus and it couldn't go from the precursor to arginine, it might need ornithine. And if that was the case, they had middle media with ornithine. Okay? So as long as there's ornithine, then it could grow. And then they had one that had citrulline, and then they one that had just arginine. So they didn't have to worry about converting anything. So they mutated the fungus just randomly. They didn't know where the X-rays were going to mutate the DNA. They weren't really like, all right, you X-ray gene, you mutate that gene. They couldn't direct that at all. Hey, Maddie. How are you? Did your mom tell you that I saw her yesterday?
Yeah, she did.
I'm sorry. About everything.
Yeah.
You're going to be fine. But I know it doesn't feel that way. Your hair looks good. Okay. Yeah. Hey, there's always a Anyways, okay, so they mutated the fungus, and then he's like, all right, I'm just going to grow them on these different media and see what happens. And he didn't, now notice on this picture, and probably in your book, there's these different types of mutants. They didn't know that at the time. They just didn't know. So they just grew them all in these different types of media and saw what happened. Well, some fungi, the wild type, and he knew the kind he didn't expose to X-rays, they grew on all the media. And it's because they had all those enzymes there, but it could grow everywhere. All right. Some of the fungi could grow on the citrulline, ornithine, and arginine media, but it could not grow in the mineral media. Well, why is that the case? It just can't grow there, but it can grow in all the other cases. Well, it's because it's probably missing this first enzyme that converted the precursor to ornithine. As long as it was given ornithine, then it could live and be fine, right? So he called the ones that could not grow in the precursor but could grow in everything else, he called it class mutant 1. The first gene in the pathway was mutated. It was the class 1 mutant. The class 2 mutants were the ones that could not grow in mineral media, could not grow in the ornithine media, but could grow in the citrulline and arginine. And he said, all right, well, if they can't grow in either one of these, but they could grow in these, then it must have the enzyme that converts ornithine to citrulline. That enzyme must not be non-functional. So that gene must be mutated. And then the class 3 mutants had the last enzyme mutated because they could not grow in any of the other environments. They could only be grown when they're actually given the arginine. Okay, so this experiment sort of supported this whole hypothesis that each gene codes for one enzyme. Because here they mutated these specific enzymes because the specific genes are mutated and then they couldn't grow or could grow, whatever. Does that make sense? Okay, I'm going to show you a video on this, and then we'll move on. Is this not narrated? All right, that's ridiculous.
No, he'll only have he'll have communication where.
So sad. Oh, never mind. It's not narrated, it'll be boring. Let's move on. Anyways, all right. So the problem with that hypothesis actually is that's not true. I'll turn that down now. Yeah, stop talking. All right. So the problem is not all proteins are enzymes, but all proteins are coded for by genes. So then what can we say? It's not one gene, one enzyme. It could be one gene. No. One gene, one protein. Okay, you gotta follow me here. So if that's the case, the problem is that a lot of proteins are like components or composed of multiple polypeptides, like hemoglobin. Made of 4 polypeptides. Collagen is made out of 3 polypeptides, and each polypeptide has its own gene. So then, what can we say? One gene, one polypeptide. Okay, we're getting closer. The problem with this one is that a lot of genes only code for RNA. They don't like tRNA or rRNA or something else, siRNA, right? So then what do we say here? One gene, one product, either RNA or a polypeptide, maybe. There's a problem with that. Is that sometimes genes can code for more than one product. Sometimes genes can code for more than one polypeptide. So this becomes problematic. So here's a quote from a paper that was from 2003, but defining a gene is problematic because one gene can code for several protein products, some genes only code for RNA, 2 genes can overlap, and there's lots of other problems. So my point in all this is that it's complicated to define what a gene actually codes for. You need to understand what's kind of going on, and you can kind of talk about it intelligently. But genes can code for RNA or polypeptides, and sometimes they can code for multiple polypeptides due to something called alternative splicing, which you learned about in this chapter, right? You read about, maybe whether you actually learned it or not is another thing. So you need to know something called the central dogma. Now, I'm going to sit here and talk at you for like the rest of the class, so just turn your brain on and don't go to sleep. Okay. Hey, these words. You learn about transcription and translation. All right, so replication, again, only happens before a cell is going to divide. So we're going to kind of put that over here. It has nothing to do with the central dogma. The central dogma of biology is that DNA goes to RNA, which goes to protein. That's the order. Okay, so DNA is transcribed into RNA, and RNA can be transcribed into polypeptides. So transcription is the process of going from DNA to RNA, and translation goes from RNA to polypeptides or proteins. So where are the genes in a bacterial cell? In the cytosol, right, in the nucleoid region, or the TANA, right? Where are the genes in a eukaryote? In the nucleus. All right. So where are proteins made in both eukaryotes and prokaryotes? Where? Where are proteins made? At the ribosomes, which are located on the cytosol. Chris, tell us, where are proteins made?
Proteins are made in the cytosol.
Yes, okay, right. Most of them. They could be attached to the ER, but they're made in the cytosol. So are ribosomes inside the nucleus, is my point in all this. No. So what has to happen? Does the DNA leave the nucleus? What leaves the nucleus? The RNA. Okay, good. Great. So something called mRNA or messenger RNA is basically like the blueprint. So if you use the analogy, the DNA is sort of like the architect. Okay, he's got the idea for how he wants to make a building in his head, right? But he just can't go to the builder and be like, I'm going to stream it to your head. It doesn't happen like that. He has to put in some sort of program and print out the blueprints or draw it, right? He's got to put it there with all the calculations. Then he hands that to the construction people and is like, here, do this. Make sense? So the architect, the dude with the idea, is sort of like your DNA. the ideas just don't come out of his head. He's got to write them down onto like the mRNA, and the mRNA leaves like the building plans and goes to whoever's going to build the building. Make sense? Okay. So let's talk a little bit about RNA. RNA is a single-stranded polynucleotide. Okay, it's made out of nucleotides. The nitrogenous bases are adenine, guanosine, and cytosine. Instead of thymine, there's uracil, which is different. You need to know that. Like I said, it's single-stranded, so there's only one strand of polynucleotides. It doesn't have two. There's different types of RNA. mRNA is messenger RNA. tRNA is transfer RNA. rRNA is ribosome RNA. siRNA is called silencing RNA. There's actually lots of types. You need to be familiar with definitely the first three on there. And we'll talk about siRNA as well. So, RNA is thought to be the first genetic material that actually evolved. We'll talk about that later on. But the reason is because it has sort of multifunctional ability. It also can function as an enzyme. We talked about it, I think, a long time ago, but there are things called ribozymes that actually RNA, they have catalytic ability. So early genetic material is thought to be RNA, one, because it's a little bit simpler, single-stranded, two, because it can have multifunctionality and do different things. So, let's talk about it later. So the process of transcription. All right, so there's three different, now this is going from DNA to RNA. Okay, you with me? So the couple things. So we're looking at our picture here. I have a big one now. Our DNA is the blue, and our RNA would be the green, and then there's only one enzyme involved in transcription. It's called RNA polymerase. There's only one. It does the unwinding of the DNA by itself. It does the building of the DNA by itself. It does everything by itself. Okay, so what happens is that the RNA will come in and recognize the beginning of a gene, where it needs to be transcribed. It will recognize there's a certain sequence. Okay, it'll recognize the time, so we'll talk about it. And it'll go along the DNA molecule. It only reads one side. Okay, the transcribed strand is called the template strand. So in this case, this is the template strand down here. The other strand that is not transcribed is called the coding strand. I want you to look at the coding strand and look at the mRNA. Tell me how they're different. The U's and T's, but everything else is the same, right? Why does that make sense?
They're a different size.
Yes, as is the same 5 to 3, and then what else? What do you think?
They're both complementary to the.
They're both complementary to the strand that's being read, right? Totally makes sense. Okay, good. Except for there's T's and the coding strand, there would be T's here. Good. And so we call this, it unwinds the DNA in about 20 nucleotides at a time. Just kind of moves around. It's called a transcription bubble. It's really small, and the DNA just winds back together. It's not like a giant replication bubble. And like I said, RNA polymerase is the only one. RNA polymerase does the same thing. It builds 5 to 3. It doesn't need like a primer, remember, because it can build by itself. RNA polymerase is the same thing that's used in DNA replication to build a primer. Anyway, so it builds 5 to 3 because it still needs that hydroxyl group to add the new nucleotides to. And then it'll stop. So let's talk about, there's different stages of transcription. Initiation, elongation, and termination. Yeah, so there's 3 stages. The only stage that's really different in eukaryotes and prokaryotes is the initiation stage. So the initiation stage in prokaryotes, you see the archaea bacteria and eubacteria, bacteria. So what happens in initiation is that the RNA polymerase will bind to something called the promoter sequence. The specific sequence of DNA nucleotides that initiates like, hey, bind here, this is the beginning of the gene. You're about to be at the beginning of the gene. It does a couple of things. It tells it where to bind. It tells it which strand of the DNA is the template strand. So which one you're going to need to copy. Okay? And so it also sort of orients it to the proper direction. So we'll always read the DNA 3 to 5. It always builds 5 to 3. Same in DNA replication. And that's initiation, and it's actually pretty easy. Yeah. The next stage in bacterial cells, prokaryotes, is elongation. What happens, the RNA polymerase will just move down, It'll unwind, like I said, around 20 base pairs at a time on the DNA, and it will just build the mRNA, the messenger RNA. And as the mRNA is being built, it sort of shoots out the end like a strand, it just comes out. The DNA will wind back together, and it'll move along, and it just goes, okay? That's basically all. There is no proofreading at all. So what happens if there's an error? Actually, no, let me tell you why. The reason why it doesn't really matter is because usually when a gene needs to be transcribed, it's going to make many copies. Let me go to another.
Is that like the RNA polymer, well, if they find a mistake, they take it out.
No, there's no proofreading ability at all. So there is no way to fix the error. But if the error is just in the RNA, it doesn't matter, as long as the error is not in the DNA. Okay, that would be a problem. If the error is in the RNA, like the RNA polymerase made a mistake, it doesn't matter. Because usually it's going to make hundreds of copies. That same gene, if it's needed at a time, that same gene is going to be transcribed like hundreds of times. You'll make hundreds of mRNAs. Most of them are going to be okay. So if they get translated, then you're going to have plenty of proteins just fine. It doesn't really matter. The mRNA doesn't live for it's not alive. It doesn't last for very long in the cytosol. There's enzymes that eventually will degrade them. They have sort of a lifespan. So it's okay. It doesn't matter. Does that make sense? Okay.
It doesn't make up when it like makes the amino acids.
What?
Doesn't it kind of make up for that when it makes the amino acids? Because like you have two different possibilities of
the wobble, you mean? Is that what you're thinking? Maybe. It depends on where the error is, actually. We'll talk about it later. It's usually fine because you're going to make a lot of copies. All right. This is another picture, I think, the one that's in your book. And this is showing, notice the bacterial cell, the DNA is just hanging out in the cytosol. Okay? The RNA is made at the same place where the ribosomes are located. So a lot of times, actually, once the RNA is transcribed, it's immediately translated at the same time. Termination happens when the RNA polymerase gets to a region on the DNA that's called the termination sequence. What happens is that it causes the RNA that's synthesized to form something called a GC hairpin turn. The RNA, as it's being synthesized, will actually base pair with itself and form this little hairpin is what it's called. And that little hairpin kind of sticks up and gets in the way and causes the RNA polymerase to fall off the DNA. And that's the end. It's really easy. That RNA in prokaryotes is done. It's already ready to be translated. Prokaryotic DNA, it's all, there's no junk DNA in bacterial cells at all. So it's ready to be translated right now. And that's it. Eukaryotes is a lot more complicated. All right, so let's talk what I'm talking about, the difference between prokaryotes and eukaryotes in terms of DNA. So prokaryotes, like I said, the DNA is in the cytosol. It's a circle, okay? There is no junk DNA at all. It's all sort of worthwhile. And the DNA is what we call naked. There are no histones. That's prokaryotes. Eukaryotes is very different. Okay, the DNA is in the nucleus. They're all straight lines. It's linear. There's a ton of junk DNA. In humans, around 99%. And the DNA is wound around proteins called histones. So that produces a few problems that have to be rectified in eukaryotes during transcription and before the RNA can be translated. A couple of things. Introns versus exons. When you think of the word exon, you should think that's the part of the RNA that needs to be expressed. Exon equals express. Okay? The introns are analogous to the junk that needs to be removed. Okay? Does that make sense? Introns are the junk, but exons equal express. Are you good? Okay. When I first learned this, when I was a freshman, in high school, I was super confused because I thought exons mean exit. And I got them totally backwards. So don't do that. Exon, express. Okay? You need to keep this. All right. Now, in eukaryotes, there are 3 different RNA polymerases that we know of. RNA polymerase 1 transcribes the rRNA, ribosomal RNA, and what's that used for? To make ribosomes. Used to make ribosomes. RNA polymerase 2 is the one we're interested in. It's the one that produces the messenger RNA. And RNA polymerase 3 also does the ribosome RNA. That's one of your books. It doesn't really matter. So, before I talk about this, I want y'all to just listen to me. Don't write anything down. So, comparatively, eukaryotes have a lot of DNA. We have 21 pairs of chromosomes in our nuclei, 46 chromosomes. All right, at any given time, we might need to transcribe, gene X, right? But it's not necessary all the time. Okay, sometimes you need more. Let's see, what do you need sometimes? Not all. More hemoglobin base. Okay, so your RNA polymerase will go in there and transcribe the hemoglobin gene. But what if you have enough of it? You want to transcribe it when you have plenty of it? No. Okay. And RNA polymerase, there's not like a different RNA polymerase for all the different genes you have. RNA polymerase should transcribe any of your genes, any or all of them. Okay. And if it goes into the nucleus and it just binds to an initiation site, it can transcribe anything. Okay. So we need to make sure we don't transcribe genes that aren't necessary. Right? Remember how we talked about the end of the last chapter, the euchromatin versus the heterochromatin? Remember some DNA is like super wound up, and even if RNA polymerase is there, it can't get in there. So at any given time, there is the euchromatin that's like sort of loose and ready to go. But even as not chromatin, sometimes we don't want to transcribe those genes, like right then. So there has to be an extra step of sort of protection to prevent genes from being transcribed when they're not needed. Okay, and that's because we have so many in eukaryotes versus prokaryotes. And that's where this comes into play. What happens in order to initiate transcription in eukaryotes is a lot more complex. It involves a lot of other proteins that need to be present before RNA polymerase will actually bind. These proteins are called transcription factors. Okay? So before RNA polymerase will actually bind to an initiation site, these transcription factors already have to be bound there. So what happens is that at the beginning, in eukaryotes, at the beginning of any particular gene, there's something called the TATA box. The reason it's called that is just because there's a TA, TAA, like a specific sequence of nucleotides, not because of anything else. But the first transcription factor will bind, and these are proteins, will bind to the TATA box. And then that will initiate other transcription factors to bind around that initiation site. You see them? They're bound around these other purple little blobs. Okay? Only then, when the first transcription factor is bound to the TATA box and all those other proteins are bound, only then will RNA polymerase come, and only then is it able to bind to that initiation site. And when that happens, you form something called the transcription initiation complex. And at that point, the gene is ready to be transcribed. RNA polymerase will not bind to the promoter region if there is no transcription factors already present. And that helps prevent transcription of genes that aren't necessary. Does that make sense? Okay. All this doesn't happen in bacteria because they don't have that many genes. It doesn't really matter. Okay. That's really the only major difference in prokaryotes versus eukaryotes is this. Initiation and elongation are basically all the same. Okay? You want to see some videos first? Yes. I'll talk about the next slide. We can go, and then I'll show videos. All right. So now there's some other differences. Like I said, because there's a lot of junk DNA, there's going to be junk RNA in the messenger RNA. So let's look at this picture here. Just look at it. Don't look it up here. Down here at the bottom, here's our DNA. The introns, the exons, the ones that we care about, are the ones in blue or gray, whatever that color is, silver. And then the introns is the junk. And notice how the junk DNA is everywhere inside of the gene. It's all over the place. It's sort of like you took your stack of cards and you just threw it up in the air, and the junk kind of landed everywhere in the middle of it. Okay? So it's not in one concentrated area. So when you do your mRNA, there's going to be the junk interspersed all throughout it. So in this picture right here, the stuff we want, the exons are going to be analogous to the green portions, and the introns will be the orange. The introns have to be taken out. We call that splicing. Before the messenger RNA can leave the nucleus, the splicing has to occur, where all the introns are taken out and the exons are all spliced together. You see down here where the green's all together now? Okay. Two other things have to happen before the mRNA can leave the nucleus. The addition of something called a poly A tail and the addition of a modified guanine cap. The poly A tail is added to the 3' end of the RNA, and that helps to protect the mRNA from degradation from enzymes in the cytosol for a little while. Here's our poly A tail. And the modified guanine cap is added to the 5' end. And it also helps a little bit of protection from degradation of enzymes in the cytosol. The modified guanine cap also serves as like a first tier sign for the ribosomal subunits to bind to before translation can happen. And they think the poly A tail aids in the exit of the mRNA from the nucleus. Helps it leave somehow. Okay, now I'm going to show you a video. Or two with it. Did y'all learn some of this in Bio 1? You did. Okay. All right, so I'm going to show you a number, so just of animation, so just watch.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
Say what? Nothing? Nothing? Yeah? What? Okay.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
Okay, real fast. Where is the energy coming from? The nucleotides come as nucleosides. Remember, the same thing is happening here, just like in DNA, right? So they come in with 3 phosphate groups, and as the 2 break off, that's where the okay. Signature.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus.
Right, that's almost ready for now. Did that help, Ish? Transcription is pretty easy. Okay, let's talk about spliceosomes. So, the splicing involves things called spliceosomes. You need to know about these guys. That's how you spell them. A spliceosome is basically a bunch of proteins and things called SNRPs, small nuclear ribonuclear proteins. And these little SNRPs, look at it. Here's a SNRP right here. Here's a protein and then a piece of snRNA. Is it RNA? Okay. Sometimes RNA can base pair with itself, which is produces those where it looks like the T. Anyway, so these come together to form a spliceosome. The function of the SNRPs is to recognize the beginning and the end of the intron. Like it'll base pair to them. Okay? And it also has, this is where you have the ribozyme activity. Those little SNRPs will actually cleave the phosphodiester bonds between the exon and the intron here and cut it. And they will also put back together the exon pieces. Okay? That's how it works.
What happens to the electrons?
It is degraded, like into their nucleotide form. Yeah.
Well, it's just there's 3 cells, and certain cells that can, that's expression.
They will produce different SNRPs when they're needed. Yes, yeah. So, and like I said, they will have their catalytic RNA and all that. We're talking about all this stuff. Now, introns are actually important because depending on the type of polynucleotide or nucleotide you're trying to make, sometimes what can be used as an intron to make like protein A can actually be treated as an exon to make protein B. Remember how I talked about one gene can code for multiple polypeptides? Are y'all actually listening? Because a lot of you aren't looking at me. They can be treated differently, and so it's called alternative splicing, and you do know about it. Basically, alternative splicing is when you can, depending on the type of polypeptide a gene is needed to make, it'll treat intron as exons in different cases. So that's how you can make different polypeptides. You didn't mean to hit my door. So, all right, now we move on to translation. Transcription is pretty easy. Translation is going from the mRNA to a polypeptide. It happens in the cytosol, utilizes ribosomes. Transcription and translation, we say, are coupled in prokaryotes because they're all happening in the cytosol, and they can actually happen at the same time. Transcription and translation are not coupled, however, in eukaryotes because you have to have the splicing that occurs first, and the mRNA has to leave the nucleus. So they occur at very different times and very different places. This is showing transcription and translation being coupled in prokaryotes. So here's the DNA strand right here. All these little purple blobs, well, they're purple down here. The little black blobs are ribosomes, and in here, the pink is showing the RNA polymerase. So notice how this same gene is being transcribed by multiple RNA polymerases at the same time. And each one of them is making a mRNA, and it's being translated by lots of ribosomes at the same time. You see that? Okay? So you can reduce huge amount of protein in just a short amount of time. So that's that. A polyribosome is basically when you have lots of ribosomes translating any one mRNA. All right, now in eukaryotes it's different. So the mRNA has to be edited and spliced and the cap and the tail added to it before it can leave the nucleus. And then it will be translated into a polypeptide at the site of ribosomes and the cytosol. So before we move in here, this is kind of the worksheet things we did last week too. We have to talk about it. So here's our DNA sequence. And here would be our RNA sequence. Can you figure that out? What is the complement to this? A and then what? U, C, G, U, Okay, good. Y'all are good, yes? Yes. All right. So what's interesting is that sequence of mRNA, let's assume it's already edited, okay, only codes for 1, 2, 3, 4, 5, 6, 7 polypeptides. 7 amino acids. But how many letters are here? 21. How does that happen? I know you know already, but we'll talk about it. We only have 4 DNA bases, but yet there are 20 total amino acids. So for a long time, scientists didn't understand, well, how does that happen? How can you have 4 bases, but yet you can make 20 different codes for 20 different amino acids? So Nuremberg and Mathai in the 60s were working on figuring out what is actually going on. The code is what we call the genetic code. And basically what they had are these little test tubes. And in their little test tubes, they had a bunch of nucleotides and their mRNAs and ribosomes and stuff like that. And they're basically like, what's going on? And they actually determined first what we call codon, amino acid match. In their test tube, they only had like uracils. That's all. They only put one type in there. So then they made a giant polynucleotide chain of uracils. And they were like, what's going on? And it's basically because they only had all these different amino acids, all of them were there. But then they made a polypeptide, and it was only phenylalanine. So if you have RNA and it's only made of uracils, you have all 20 amino acids in the test tube, and then your ribosomes and all the other stuff you needed, but you only make a phenylalanine chain. Like, why? You know what I'm saying? Mm-hmm. They had RNA strand that was made up of just uracil. They had all 20 amino acids in their test tube. They had ribosomes. They had all the stuff they needed to make a polypeptide chain. They only made polypeptide chains that were made out of phenylalanine. Why? Because the RNA only had uracil, right? Does that make sense? No, you're staring at me. Okay. Well, anyway, what you can read about in your book. It's because there are only uracils in that chain, and the ribosomes read polynucleotide chains. They read the RNA. Anyways, okay. So there they are. They did a lot of work. So basically, eventually, now we know that our code is basically called a triplet code. It's blocks of 3 RNA. The codons are actually on the RNA. Blocks of 3 RNA nucleotides together is what we call a codon. So they're read in steps of 3. So if we actually space it out, it's like that. And to be honest, when I do work like this, when you do transcription like the worksheets I gave you, it makes it a lot easier to draw a line like this. You don't get confused. And that's how you read it. So AUG is always what we call the start codon, and it always codes for methionine. So if you look at it, y'all know how to read that chart, obviously you do. Okay, good. There are two different charts. The one in your book is the most common, but there's also this circle chart that I have up here. I like the circle chart better too. But anyway, the circle chart, you start reading from the middle and you work your way out. Anyway, okay. So let's talk about characteristics of the genetic code. You need to know this. There's three. Okay, one, the genetic code is universal. 99% of organisms on the whole planet, 99.9 really, use the same genetic code. I can take a gene from you, like insulin, put it into a bacterial cell, and that bacteria will make insulin. That means that the genetic code is the same. So if you take your gene and put it into a bacteria and don't give it anything else except for the DNA, it can still make insulin because it still uses the same nucleotides, it still uses the same process machinery, still uses the same amino acids. Okay? A lot of genetic engineering and stuff like that has happened because the genetic code is universal. It's also a really big support for common descent. We'll talk about that later on. All right, the code is unambiguous. What does that mean? You don't know what ambiguous means?
It's like specific for each of us.
Right. So ambiguous, what does ambiguous mean?
Not clear.
It's not clear. So basically what I'm saying, the code is very clear. If I say you have a codon that's C or that's UUU, what's it going to code for every single time? Phenylalanine. You can find on it if you don't remember, I just showed you, but where's our UUU? Here it is. Okay, what if it's like AAA? What's it going to code for? Every single time. LYS, which is lysine. Okay? It is not ambiguous. Every single codon has a specific amino acid it codes for, a specific thing it's coding for. You also say that the code is redundant. What does that mean? Do what? Yeah. Yes. There are multiple codons that code for almost all of the amino acids. Look at that proline right here. There's 4 different codons that will code for proline. You see that? Yes? So y'all listen to me. Y'all sort of bombed the last test, and I'm sort of nervous about the next test. But it's not my job to study for your test, right? If you're sitting in class and you're on your cell phones, which a lot of you are, you're reading other books, which a lot of you are, you're doing things on your computers, which I can even tell what you're doing, and you're not paying attention, whose fault is that? Not mine. Okay, so I'm not going to feel sorry for you at all on these tests anymore. You need to step up, or else you're not going to pass the AP exam at all. Okay? Anyway. And if you're going to come to class and be on your phone the whole time, just don't come. It gets really sort of annoying. Okay? I can just sit here and give you worksheets and tell you to just learn stuff on your own. I'm just saying. Anyways, it's redundant, meaning it codes for the same thing. So, if, the only one that actually will code for only one thing is what? There's one codon that will only code for one thing. The start codon. And it's actually really important there's only one codon that is the start. Because you don't want to start translating it multiple parts. The start codon needs to be the same for all the time, so you only start where it needs to start. Anyways, AUG is the start codon. I'm going to tell you on every test, on everything, you're going to be given a codon chart like this. You'll be given this, and it will tell you everything. It'll say that stuff, okay? There are also 3 stop codons. 3 different ones, UGA, UAA, and UAG. And there they are. And they don't actually code for amino acid at all. They just tell the thing that causes everything to fall apart, and I'll tell you how that works later on. Okay, now you won't have to memorize what those abbreviations stand for, like LEU is leucine, this is isoleucine, valine, alanine. You don't have to memorize those. I know those because I've taken a lot of other classes. You just have to know you just have to know what it means. Those are amino acids. All right, there's a start and the stop codon. Now, this is just an example of the code being universal. Here's a tobacco plant that's actually expressing a gene from a firefly. So genetic engineers put a firefly gene into the tobacco plant, allowed it to grow, and it's making that same protein that actually fluoresces under like the black light, and there's going. Here's a pig that's showing the GFP gene, green fluorescent protein gene, in its specific tissue that make up its nose and its hooves, and you can see it's fluorescing. It's just showing, like, they didn't do this for any reason. It's not hurting the animals. This is not ethical to do to humans, I'm just saying. Because you can't let an embryo choose whether it wants to be fluoresce or not, so you can't tell it. You just wouldn't do that, I'm just saying. You could, but we don't. The problem is we can't ask you before you're conceived, huh? Right, you know.
Would you like to?
I need to be cool, but. All right. So, were y'all okay on all that work I gave you last week? Those worksheets where you had to like do it yourself. That's the basis of the lab for the next 2 days, so I don't really want to spend a lot of time on like the process right now. I just want to talk about how it actually all works. So the process of translation. How in the world does the ribosome know like what amino acid to put where? Does anybody know? It involves something called transfer RNA. Transfer RNA is an RNA molecule that exists inside cells, and I'm going to just get to it. It looks like this in reality. It's like a clover leaf, and it's base paired in certain regions. But at one end, it has the site, the 3' end, where an amino acid will attach itself. Okay? These other sites on here, the blue and the pink, and then down here at the bottom, the green. The blue and the pink allow the tRNA to bind, hydrogen bond, to the ribosome when it gets into the different sites. And at the bottom, we have something called the anticodon. The anticodon is complementary to the codon. Put that. So here's the start codon, AUG. The anticodon will be UAC. It's complementary to the codon. You see that? Okay? That means it'll base pair to the codon, and only will base pair to a specific codon. Okay? And it comes with bringing its little amino acid, which in this case is methionine. And it allows only for one type of amino acid to be added to a specific codon. You see that? Okay? So the anticodon for CGU would be GCA. What's the anticodon for GUA? Right. Notice how it compares to the DNA template strand. It's basically the same except for where there's C's, there's U's, right? Okay. Y'all understand the anticodon? A lot of times people get confused on what that is. Okay. Now in your book, they're going to show it like this little thing here. In reality, it's kind of space fills and folds all on itself, but here we go. So in your book, the green little guy, here's the anticodon on the bottom, and the top is the amino acid attachment site. The tRNAs aren't degraded, they're reloadable. So basically once it drops off one amino acid, it can go back and go pick up another one and be reused over and over again. Yeah?
What's the point of the missing bases in the left picture?
Missing bases.
Like, in the anticodon, there's one missing. Is there fluorescent? Like at the bottom? Yeah.
Oh, it's just showing there's space there. It's not really missing anything. Don't worry about that. Don't worry about it. Nothing. Yeah.
So each tRNA only has like one set of anticodons?
Each tRNA only has one anticodon. Yes. Yeah. Yeah. There's not multiple. Okay. Now, how many amino acids are there? How many tRNA do you think there are? Okay. There's a lot of tRNAs, but there aren't 64. There aren't, like, how many different codons? Now I'll go back to our codon chart. Okay. So each one of these codons codes for something. If you think about it that way, you would think there would be how many different tRNAs? 64. There aren't. And it's because something called the wobble effect. If you actually read in synthesizes, you remember this maybe? The wobble effect. A lot of times in the tRNA, in the third part of the anticodon, that third nucleotide, it's a specific nucleotide that can do what we call flexible base pairing. It can base pair with multiple types of nucleotides. So look here. Does it matter what the third nucleotide is in the anticodon for proline? Does it really matter? Not at all. Because as long as it's got a C and a C, the third one doesn't matter. It's all coding for proline.
Yeah, that's what I was talking about earlier, with like the self-correcting thing.
Oh, yeah, that's what I thought you were talking about, but, right. But it has to be in that third spot. Okay, so in reality, there's not 64 tRNAs, there's actually a lot less, and it's because of this wobble effect. Okay, so even that threonine, you can have just one, that, as long as it's an A and C, it doesn't matter what's in the third spot. So y'all read about that. And this is just showing that tRNAs are reloadable. There's an enzyme called aminoacyltRNA synthetase that hangs out in the cytosol that will take in a tRNA that's what we call empty. Doesn't have an amino acid. It'll take in an amino acid, uses ATP energy to bind the amino acid to the tRNA, and now it's ready to go to a ribosome. And drop it off and does the whole thing over again. Yes, and there's the wobble. Now let's talk about ribosomes. This is really dense, isn't it? Sorry. All right, so ribosomes are made up of two subunits, a large and a small. I think this whole thing reminds me of a poop or something. Anyways, there's large and small subunits. The small subunit is on the bottom, the large is up here. And in between them will be the messenger RNA. In the large subunits, there are three areas. We call the E, the P, and the A. E stands for exit, P stands for peptidyltRNA binding site, and A stands for aminoacyltRNA binding site. We'll talk about all of them. This is what a space filling model would actually look like. All right, so I'm just going to show you all this stuff in the picture. So there's three stages to translation: initiation, elongation, termination. They have the same names as transcription. They're very different. So let's talk about what happens. So initiation. The small ribosomal subunit will come and bind to the 5' end of the mRNA. How does it know where to go? That modified guanine cap's hanging out over there, saying over here, okay? It'll kind of orient itself till it gets to the start codon area, okay? Now there's other proteins that help in this called translation initiation factors that aren't shown in these pictures for the sake of like not confusing the heck out of you. Okay, so just know there's other proteins involved, they just don't show them in any of these pictures. So the small subunit will come in and bind. And then what happens is that the first transfer RNA will come and bind to the start codon. That happens first. And then using the energy of GTP, the large subunit will come and orient itself on top of all that, such that that first mRNA, or the first tRNA, is in the P site. See that? So it's like the small subunit's already there, the first tRNA is already there, and then the large subunit comes and like sits on top of it. Such that that mRNA, the tRNA, is in the P site, in the one in the middle. See that? That's initiation. It's done. So then we do elongation. I'm just going to talk about it using pictures because it's easier to understand. So how long has this been happening? A little while. See how there's 4 amino acids here? Okay, here there's just one. Okay, so this has been happening for a little bit. So here we have our tRNA in the P site that has the growing polypeptide chain. Then the next tRNA comes into the A site. Now, does it know immediately it's going to actually fit? No, it might come and bounce out. And some of the animations I'm going to show you, they're going to be coming in and out, coming out. But if it has the actual anticodon, it will base pair with the codon. It will kind of sort of stick there. Okay? And if it does that, if it sticks there, notice it's stuck there. There we go. So then what happens is that using the energy of GTP, this red arrow happens. See that red arrow? The polypeptide chain on the tRNA in the P site breaks off of this tRNA and binds, makes a peptide bond to the amino acid on the tRNA in the A site. Notice the difference between this picture and that picture. You see? Okay. Now, what do we have to do next? This little guy's finished. He's done. Yeah. Yeah. Basically what has to happen is this tRNA needs to go to the E site, this one needs to go to the P site, and the A site needs to become empty again so we can do it all over again. So what happens, so let's imagine this is our mRNA in the middle and then our ribosome around it, okay? It's not like the mRNA, like, kind of, the whole complex, like, moves. Like, the distance of one codon, okay? It's sort of like clicker tape, kind of goes through. So see the difference? So it's like the mRNA moves, but this all kind of stays together. So it kind of goes the distance of one codon, such that the tRNA that was in the P site is now in the E site. Now let me go back for a second. Over here. You see how the E site is a little bit shorter than these two sites? Okay. So to show that, because once the tRNA gets in the E site, it won't fit very well. It's like, it sort of doesn't fit and then it just falls out. It's kicked out, basically. So once it gets in there, it's kicked out, basically. And then we can do the whole thing over and over again. And this whole thing will happen over and over and over again until we get a stop codon in the A site. Yeah.
So technically, it's not that the tRNA is moving,
Yeah. Yeah. The tRNA stays hydrogen bonded to the mRNA. And it just stays there. Yeah, and it's just like the whole thing kind of shifts around it. Yeah. Good question. And this will keep happening until we get a stop codon.
Ever like somehow the tRNA doesn't make it to like the codon and like it's
I mean, I guess it could happen, but it will just stay stuck there until one eventually gets there. No. All right, so a stop codon doesn't actually code for anything, but there's a thing called a release factor that will come in and bind here, and that release factor causes sort of a chain reaction to happen. So a couple of things happen. One, it adds a water molecule to release the polypeptide chain from the tRNA. And then two, energy is used to sort of break off the ribosomes from the mRNA. And it all falls apart. And that's the end. And the polypeptide is now, it'll go fold up and do what it needs to do to associate with another one or whatever. And that's the end of translation. I will show you some animations and I will finish these notes tomorrow. We still have a little bit to do. Not a lot. Pretty dense, I know. All right. Yeah.
What's the polypeptide release?
What does the polypeptide do?
Yeah.
It'll fold up all automatically and either associate with another one to help make like hemoglobin, or it might be a fully functional protein as well and just do whatever it needs to do.
You put fold inside the cell or is that outside?
It folds in the cytosol automatically. In some of the animations, I'll show you see it folding up. It might show it here, I don't remember.
The messenger mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the larger subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated.
Now, do you hear how she's saying the ribosome helps to form that new bond? It's because in the ribosome, there's our RNA, that ribosomal RNA, helps to do two things. One, serves as an enzyme to help catalyze these peptide bonds forming, but it also helps to base pair to the tRNA to hold them in these sites.
Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
All right, I'll see you all tomorrow.